Katie received her Bachelor's of Science in Forest Health from the State University of New York College of Environmental Science and Forestry in Syracuse. Her Master's and PhD were completed at WSU in Puyallup Research and Extension Center under the mentorship of Dr. Gary Chastagner, sorry if I butchered that name. Her research focused on root rot species affecting western forests and Christmas trees. And today she's currently the forest pathologist with the Montana Department of Natural Resources and Conservation Forestry Division in Missoula, Montana. I'd like to note that this webinar is co-sponsored by Montana State University Extension Integrated Pest Management, and I'm really happy to have Katie here with us today. So I'm going to turn it over to her, and we will get started. Just a quick reminder, put your questions and answers in the Q&A window, please, and reserve the chat window for any questions you might have for me. That will help us stay a little bit more organized and move quicker and be able to address questions and answers more succinctly. So it's all you, Katie.
So my name is Katie McKeever. I am forest pathologist with the Montana Department of Natural Resources and Conservation, and I'm based out of Missoula. So I was really pleased to be asked to give this talk today on white pine blister rust because it's such an exciting topic with a really rich history. So it's really fun to talk about. So to kick things off here, I'll bring your attention to my picture on my cover slide. This picture was taken about a year ago, September 2017, and this was taken down in south central Montana, right around Big Sky, if you're familiar with the state. And this is just a prime example of some of the absolute devastation that we've seen with white pine blister rust, of course, in conjunction with mountain pine beetle, in some of our high elevation whitebark pine ecosystems. So all of these dead trees you see in this picture here are whitebark pines. It's pretty striking. So white pine blister rust is the name of a disease, and this is caused by the fungus Crenartium rubicola. This fungus affects 5-needle pines, so those are pinus species that hold their needles in bundles of 5, as opposed to some other pines that hold them in bundles of 2 or 3. This fungus also affects some shrub species like currants and gooseberries, scarlet paintbrush, and some louseworts. This fungus is introduced, so it is not native to North America. Its native range is in central Siberia, sort of east of the Ural Mountains, even extending down into Asia. And this was introduced around the turn of the century. So for the last 120 years, this fungus and this disease has influenced timber industries, it's inspired generations of plant pathologists, and it's spurred some really interesting innovations in things like resistance breeding in trees. So Crenartium rubicola is a rust fungus. Rust fungi are an incredibly diverse order of fungi, which includes thousands of different species, affecting nearly every family of plant. So we have rusts on grasses, forbs, trees, shrubs, you name it. There's a rust fungus that affects it. They are obligate parasites, which means that they need their hosts to be alive for them to survive and complete their life cycle. They are also biotrophs, which mean that they invade and derive their nutrition from living cells. And this is in contrast to those fungi that exude toxins that actually kill the cells in advance of their colonization. They have incredibly complex life cycles, so they can have as many as 5 different spore stages, which we call macrocyclic. They may have as few as 2, which we call microcyclic, or somewhere in between, which is called demicyclic. They often require 2 unrelated plant species to complete their life cycle, which we call heteroecious. There are some rusts that only require 1 plant to complete their life cycle, which we call autoecious. But Crenartium rubicola is a macrocyclic heteroecious rust. So with heteroecious rusts that require 2 hosts, we typically refer to these hosts as principal and alternate hosts. Principal hosts typically suffer a little bit more damage. They're often killed. They're usually a little bit more economically or ecologically important. Alternate hosts usually suffer mild symptoms and usually aren't quite as economically influential, although they do very often have horticultural value. In the white pine blister rust system, the principal hosts are those 5-needle pines, of which we have 7 species in North America, including important timber species like eastern and western white pine and sugar pine. Also some ecological keystone species, things like limber, whitebark, and bristlecone pines. It was thought for nearly a century that the only alternate hosts we had in this system were gooseberries and currants, so plants in the genus Ribes. It wasn't until 2006 that it was established that 2 other plant species could also support portions of this life cycle, including scarlet paintbrush in the genus Castanea and some lousewort species in the genus Pedicularis. So this was monumental in our understanding of blister rust, particularly in the western US. Again, on those pines, a lot of damage, often killed. We can get things like spindle-shaped swelling and deformity on those branches and stems. The bark becomes broken and blocky when those blisters burst through and break the bark year after year. Top and branch kill when the branches and stems are girdled by the fungus. Heavy resin flow at the site of infection. Also, some of these spore stages apparently are sweet in flavor, and that thickened bark in the area of infection is attractive to rodents. So we get a lot of rodent gnawing on the trees where the infection occurs, which is really damaging to these trees. In contrast, on the alternate hosts, we see typically mild symptoms, things like leaf spots. They might have early leaf drop or abscission. But these hosts actually support the bulk of the life cycle of the fungus. So 3 of those 5 spore stages are produced on these alternate hosts, occurring in clusters of spores on the underside of the leaves. So we do have to address some terminology here for an intelligent discussion of blister rust. We have to tackle some pretty gnarly words. As a macrocyclic rust, Crenartium rubicola utilizes all 5 of those spore stages. So common notation for macrocyclic rusts is to denote the spore stages with Roman numerals, starting from 0 to 4. And usually the pycnial stage is at 0, so that's where we'll start. So at 0, I have pycniospores. These are produced on pine, as you can see from my leaf outlines here. The pycniospores have complementary mating types, so we can think of these as genders, male, female, plus, minus. But 2 of these mating types will fuse, and they will give rise to aciospores, which is at Roman numeral 1, also produced on pine. These are the only spore stage that can then go and infect the alternate host. So moving over to my alternate host leaf at Roman numeral 2, uridinospores. These are the repeating spore stage. So these spores continuously reinfect this host throughout the growing season. And we'll address that a little bit more in a second. Teleospores, also produced on the alternate host, give rise to basidiospores, which are the only spore stage that can then go back and infect pines. And to make that a little bit more clear how this disease spreads, I found this neat little federal publication from the 1950s that lays it out pretty nicely. Blister rust does not spread directly from pine to pine. The spores that are produced in the pine spread first to the alternate host, which may be Ribes. It bounces around on the alternate host there for a growing season, and then the spores produced in the alternate host spread back to more pines. So let's talk about the disease cycle here, getting a feeling for how the spores look and how this fungus moves through the hosts. Here I'm going to start over on the left-hand side of the slide with basidiospores at A. And I know we talked about the Roman numeral notation starting with pycniospores, but it's easier to describe the movement starting at basidiospores. I also just like to confuse people, so we're going to start there. Basidiospores are produced on the alternate host. In autumn, typically, they fly and they land on pine needles. Now, the current season's pine needles, the ones that just emerged this past spring, are actually pretty resistant to the fungus. So it's the 2nd and 3rd year's needles, the older needles, that are actually infected. The fungus grows right through the stomata on the underside of the needle, which are the natural openings that are there for gas exchange. So it's a very passive infection. The fungus grows through the needles, grows into the twigs, into branches, and ultimately back into the stem. So moving over to the top right-hand side of the slide, over at pycnospores. These are produced in reddish droplets of ooze or nectar, and these are produced in the woody tissues where that fungus is residing, and the area that's colonized where that fungus is, we call that the canker. So you'll hear that term referred to quite a bit. These may occur as soon as the first growing season following initial infection of the needles, but the fungus may lie dormant. There may be a latent period of up to 2 or even 3 years before you see pycnospores. Again, they have the opposite mating types. They fuse, give rise to aciospores. They're a C. Aciospores are produced in those blisters that burst through the bark and give this disease its classic look and its name. Those blisters are covered by a thin membrane. That membrane ruptures, and it releases these dry, dusty spores out into the air. They're carried on wind currents where they fly and they land on the alternate host. So down in the lower right-hand corner at D, I have a ribes leaf there. The aciospores land on that leaf. They can infect again through the stomata or by brute force right through the cuticle of that leaf. And within days to weeks, they give rise to clusters of uridinospores on the underside of the leaf. Uridinospores are distributed on the wind. They may then land right back on that same plant or on adjacent plants of that same alternate host, where they infect. They give rise to more uridinospores. They land on more alternate hosts, give rise to more and more, so on. So this is that repeating stage. And they will continuously reinfect this host throughout the growing season, which serves to increase the amount of fungus that's available for later stages of disease, so increase inoculum. And it also spreads the disease spatially throughout the stand. So given some environmental cue toward the end of the growing season, these uridinospores instead differentiate into telia. Telia are long stalk or horn type structures that protrude right from the bottom of the leaf. And the teliospores are born on these telia. So over in the left-hand lower corner, I have a black and white micrograph of a telium containing teliospores. And those teliospores are the little worm-looking things on there. And the teliospores are the site of meiosis. So I won't go into the details of meiosis. I will spare you. You should remember this from your basic biology. But meiosis splits the parent genome into 4 identical daughter cells. So each one of those teliospores gives rise to 4 basidiospores, which are born on little pegs on each spore. I have a red arrow pointing to one of those in that black and white micrograph. These basidiospores are then dislodged by air or rain splash, where they then go back to land on pine needles and complete the life cycle. So the next plausible question to ask is, how does this fungus, these little sensitive spores, how do they get all the way from somewhere like Siberia to begin causing problems in North America? Well, humans, of course. I don't know that anybody's terribly surprised by that. We kind of have a knack for moving things globally and introducing things. And this began a long time ago. So prior to 1700 even, Europeans were importing ribes plants for use in their gardens and for a fruit crop. And there were ribes that are native to Europe, but some of these imported ribes had nice horticultural value. So we were importing ribes plants from places like Central Siberia and inadvertently imported the Blister fungus in an unnoticeable state on some of these ribes leaves. About the same time, Europeans were also going and settling the New World. So that is, of course, the North America, particularly the east coast of North America. And when these settlers stepped off their ships, they were greeted by these big, beautiful, tall pines, which they didn't have. These pines, white pine is native to North America. So they saw these pines and gosh, they were just perfect for things like ship masts and keels. So they began cutting down white pines and shipping logs back to Europe. And upon returning home, some of these settlers would take white pine seedlings and plant them. Some of the first white pines that were planted in the UK were planted on the estate of Lord Weymouth. So eastern white pine is actually known as Weymouth pine in the UK, just a little bit of trivia there. So now we have these two susceptible plants together in a spot with a very virulent introduced fungus. It wasn't much longer before a sort of rusty color disease, an early leaf drop was noticed on some of these imported ribes, and a blistery kind of disease was noticed on some of our pines. And it wasn't until the 1880s that it was actually established that these are two spore stages of the same fungus. Meanwhile, in the US, the population was booming. So the new world was being settled by people from all over Europe and all of these other countries. So people were building houses, and they were building forts, and they were building ships. So we were cutting down these white pines at an intense rate. Increases and advances in logging technology just increased the speed and efficiency of deforestation. So it wasn't long before people realized, maybe we should begin planting some of these pines to replace some of the ones we're cutting, make this a more sustainable system. Now, there were conifer nurseries operating in the US at that time, or tree nurseries, I should say, but they were growing mostly ornamentals and fruit trees for use in people's gardens as a food crop. They weren't growing white pine. Also, we had this brand new continent fully forested, so it seemed like an endless resource, so it wasn't profitable to be growing white pine seedlings. However, in Europe, of course, they were growing white pine like crazy. It was this brand new, incredible species, and they were producing it in bulk. So to aid in reforestation efforts in the US, we got rid of tariffs on imports and began importing bulk amounts of white pine seedlings. Of course, we inadvertently imported the blister rust fungus as well. So blister rust was first detected on ribes at an experimental station in Geneva, New York around 1906. And then it was, of course, noticed in planted pines, and then, of course, in our native forests. Concurrently, we were importing white pine seedlings into Western ports also, places like Seattle and Vancouver. And around 1921, it was observed to have this disease in Western white pine populations around Vancouver. So what are some of the consequences of this importation? What have we done? Of course, it's had big impacts on timber resources. Things like Western white pine, sugar pine, and of course Eastern white pine in the East are really valuable timber species. These trees grow tall and straight. Their crowns shade out their lower branches so they naturally prune, so they have clear boles free of knots. The wood is workable, it finished really well. These trees were incredibly productive. So down in this slide in the lower center photo, I have some logs on a cart. And these logs came from what was known as the white pine king. This was a tree that was felled in 1911 on potlatch lands around Bowville, Idaho. And this tree was estimated to hold over 29,000 board feet in this tree alone. And over on the right side of this slide, we have a gentleman standing in front of some boards. These are boards cut from that white pine king. Huge boards, the width of 3 men, and beautiful wood. And talk about productive. These trees could attain massive sizes. For Western white pine in its heyday, it was estimated that a stand that was nearly 100 years old could produce as much as 50,000 board feet per acre. So just to put that into perspective, that's the equivalent of 5 medium-sized houses from a single acre of forest. And these trees didn't only live to be 100 years old, they were 200, 300, 500 years old. So from some of these older stands, we get upwards of 100,000 board feet per acre. Incredible. Sugar pine are some of the tallest and girthiest of all of the pine species in the world. These trees are endemic to the Southern Cascades and Sierra of Oregon and California. And these trees this size, the ecosystem resources that they provide, shade, moisture, nutrients, so important. When trees this big get hollowed out, the cavity nesting wildlife that can use them. Western white pine was historically a major component of the forests of the Idaho Panhandle and into western Montana. This species now sits on 5% of its original range. That's a 95% reduction in mere decades when these trees were centuries year old. Big impacts. Besides timber resources, these things are ecologically important. So trees like whitebark, limber, and bristlecone pines, these are these venerable old inhabitants of these high elevation, harsh ecosystems. Bristlecone pines are among the oldest of all living things. In excess of 5,000 years, these trees can grow. Where these trees exist on these exposed landscapes, they provide things like shelter and moisture. So when seeds from other places are blown in, they hit these trees, they fall to the base, and they have everything they need to germinate and grow. So where we get these clusters of trees and vegetation, we call this vegetation islands. And these tree or vegetation islands stabilize the soil in these windswept areas. They retain snowpack, and that snowpack retention is so important for metering our snow melt down through our watersheds throughout the summer months. This is really important in the arid West for things like agriculture, irrigation, and wildlife. The big seeds that are produced in these cones are an important seed source for a lot of wildlife. Bears, both grizzly and black bears, use these seeds. Particularly when they come out of hibernation and food is scarce, they can depend on that food source. These trees have a special relationship with this bird that's pictured here. That's the Clark's nutcracker. The Clark's nutcracker uses the tree for shelter and for food. It also takes these seeds and it'll cache them in the ground for use as a later food source. It remembers most of them, but it's forgetful and it forgets a few of these caches. And these will grow into clusters of new trees. So this bird is actually planting these trees in areas that are just perfect for their survival and growth. Besides all of these important functions, these trees just have spiritual and aesthetic value for the people that live and recreate in these areas. So how has this fungus been so successful? There are a few reasons. One is that it's invasive, it's non-native. So these trees have not co-evolved. They haven't battled back and forth with this fungus for millennia. And this battle back and forth or co-evolution, this builds resistance into the populations through natural selection. When this fungus arrived, these trees were essentially naturally defenseless. They had never seen it before. This fungus also does not discriminate on the size or age of tree that it can kill. It can kill reproductively mature trees. So those are the ones that are bearing cones. When it girdles and kills branches that have cones or kills these older trees, we're reducing the seed source that's available to give rise to the next generation. This fungus can also kill small trees. So any seeds that do germinate and grow are killed before they even reach reproductive maturity. The effects of this fungus, of course, are exacerbated by native bark beetles. So we have things like western pine beetle and mountain pine beetle that are native. These trees have co-evolved with them, but they have cyclical population booms, where for several years they'll come through and they'll leave swaths of dead trees in their path. This is a natural cycle. So these beetles kill the trees, the standing timber fuels wildfires. Wildfires come through, they leave that ground fertile. They create openings in the canopy that allow sunlight down to the ground. And these areas are just perfect for white pine regeneration. But with the impact that the fungus now has on seed source and production and regeneration and survival, these white pines just can't bounce back as they once would from normal pine beetle outbreaks. So we've actually disrupted the natural cycle of things, and the combination of these two organisms has hastened the losses of these pines throughout their ranges. So what's been done to combat blister rust? What can we do now? Because clearly we have to live with it. And what's the future for white pines in North America? So very early on, people realized this is a problem and something needs to be done. So as early as 1909, there were efforts to regulate the movement of plant material. In 1912, the Plant Quarantine Act was passed, which prohibited importation of 5-needle pines as well as importation of exotic ribes. In 1915, the USDA created the Office of Blister Rust Control, or OBRC. And OBRC appointed nursery inspectors who were tasked with going to nurseries, looking for disease seedlings and making sure that disease seedlings were destroyed before they could be outplanted. OBRC also created officers to oversee the removal of exotic rabies, including from plantings and people's homes and gardens. So this was a very contentious issue. People didn't want to get rid of their plants that they had paid for and grown. So some states actually began to offer reparations to get people to comply with these new mandates. Because some of this small-scale eradication and looking for disease seedlings appeared to be working, some of the states got together along with OBRC and tried to expand rabies eradication to landscape levels, to the wild. So this rabies eradication effort was a coordinated effort that lasted more than 40 years. It started prior to 1920 and lasted well into the 60s. This included salaried full-time employees of the OBRC. When World War II broke out, many of these men had to go to war. This position was largely backfilled with seasonal high school and college kids. During the Great Depression, Roosevelt's Civilian Conservation Corps, CCC, was instrumental in rabies eradication. And this was backbreaking work. This was incredible effort. This involved pulling, digging, and herbiciding these plants out of the ground. I have two photos on this slide. On the left, I have a man who's got a backpack sprayer on, spraying rabies plants on the side of a waterfall in Glacier National Park. On the right, a man literally hand weeding the forest. So this was hard work. The terrain was difficult. It was hot out. It was summer. These plants would resprout year after year. The herbicides they were using were toxic sometimes, and in some cases, highly flammable. This big effort, though, had a huge impact on a lot of families. It really influenced our history as a nation. It was an important source of income in lean times, especially during the Great Depression. It inspired generations of plant pathologists by bringing awareness to tree disease and forest health issues. Despite this effort, rabies eradication was largely a failure. And in hindsight, there are actually a few reasons for this. I'll name three here. One, weeding the forest is nearly impossible. The amount of acreage you had to cover, the terrain, it was almost impossible to get a man into every part of the backcountry that had blister rust, that had white pines and rabies coexisting. Also, the spores can fly much further than the radius of removal of the rabies around white pine plantings. So originally, it was mandated to have about a 100-meter radius of rabies removal around white pines. This was increased incrementally over the years. In some cases, it reached a mile of removal. But we know now with the wind speed, wind direction, topography, these spores may fly much further than the mandated removal radii, so they were inadequate. Also, we know now, of course, there are two other alternate hosts, at least two other alternate hosts that are supporting portions of the life cycle of this fungus. So despite our best efforts, the fungus was persisting. So what are we doing now? We have to live with blister rust, but we are able to culture and grow white pines, and we're doing this using integrated pest management, or IPM, which is essentially cultural methods that are supplemented with chemical controls when necessary. And one of these important IPM methods is the pruning of lower branches, particularly on younger trees. And the reason we're doing lower branch pruning is that it was noticed that the lower branches were more commonly becoming infected. And this is because down near the ground, there's a high humidity environment, which is just perfect for fungal development. Also, those branches were just in contact with the fungus more. They're down there where the spores are coming off of these low stature hosts. So to reduce some of that contact between host and pathogen, we're removing lower branches. And on very young plantings, this can be as much as the lower half of the crown. As these trees grow, it takes a lot of time and attention to come back year after year and prune as high as 8 to 10 feet on these trees. Now, this is typically recommended for trees that are younger, less than 25, one, because it's more difficult to remove older branches, but also because as these trees grow, their crowns grow and shade out lower branches naturally, so it saves us some of the work. You can also prune off active infections. So the way that this disease moves from needle to branch to stem, we might be able to prune off a branch infection before it reaches the stem. The rule of thumb here is that if that canker is greater than 5 inches from the main stem, it's prunable. If it's less than 5 inches to the main stem, it's typically regarded that that fungus is already in the main stem, just not noticeable yet, so it might not be worth the time and labor. Another important IPM method is manipulating planting. So we can choose low-risk sites. We can choose sites that don't have many ribes, or depending on the species of ribes, they vary in susceptibility. Also choosing sites with good growing conditions, because if these trees are growing vigorously, we're giving them every chance that they have to express any natural resistance they might have. So thinking about things like soil type, sunlight, freeze and frost, moisture, to allow these plants to grow well. Decreasing densities in our plantings will increase airflow and reduce that high humidity environment that's so good for fungal development. Removing any alternate hosts. If we have a deliberate planting site and it's feasible to remove alternate hosts, this is a good option. It will break the life cycle. Using seedlings that are adapted to the climate zone and the site is also really important to let them grow as well as they can. And to know where to plant or when to prune and do all of these steps, it's important to monitor your site. So monitoring and scouting are incredibly important portions of any IPM program. Another great tool we have is resistant trees. So breeding for resistance began with foresters, scientists, and other people just walking around the forests and noticing that there were large, mature trees that were apparently free of disease or had very low levels of disease. And these were in areas where other trees were just hammered by the disease. So these are potentially resistant trees. We call these plus trees. So very early efforts for breeding involved identifying a few or many of these plus trees and actually climbing them and hand pollinating female flowers with pollen from other resistant trees. And the hope here is to get cones that have seeds from 2 resistant parents. In lieu of some of these hand pollination methods, we might just also identify plus trees and climb them and collect the cones from them. We do this for whitebark and limber. We extract the seed and then we grow the seedlings. Now, to know if any of these offspring seedlings have inherited resistance from their parents, we need to inoculate them. We need to give them the disease. So down the lower right side of this slide, I have an example of how this might be done. This is a breeding program in Oregon. So they grow the seedlings in cones, and then they lay infected ribes leaves on screens over top of the seedlings and let them just rain spores down onto them. These inoculated seedlings are then planted into blocks according to family line or parent tree, and disease is allowed to develop. So many of these trees die off, they're not resistant, but many of them that survive are potentially resistant trees. These survivors may either then be further pollinated and bred with each other to produce more seed and more seedlings that are tested. They may just be directly outplanted or grafted into seed orchards where they can open pollinate or wind pollinate with other seed orchard trees to produce resistant seedlings. Now, by continuing this controlled pollination through further generations, it helps us to increase the frequency of seedlings in the offspring population that may be resistant. And it also stabilizes that resistance in the genetic code of some of these individual seedlings. Breeding programs were initiated pretty early on, as early as the 1940s in places like Wisconsin with eastern white pine. In the 1950s in Washington and Oregon, we established programs for western white pine and sugar pine. And for whitebark, limber, even bristlecone, some of these programs are currently underway. Now, to test the validity of resistance and also to let these trees get natural infections and also just to look at their horticultural characteristics. So how well do these trees grow? We take some of these improved seedlings and we plant them into operational landscape plantings, and then we follow them for many years. It's still natural to expect losses, even in these improved seedlings, because there's a lot of impact from environment there. So if these trees are under stress, they might not express the resistance as well as they would under controlled conditions. The rule of thumb here is phenotype, which is in this case resistance or our characteristic of interest, is controlled by the genotype of the host, as well as input from the environment. So things like moisture stress and heat and crowding can all affect how well resistance is expressed. We might even have two seedlings from the same family line that are just outplanted into two different areas, and they'll have very different reactions. So it's very complex. Despite losses, though, the survival of improved seedlings is far superior to anything that we see in wild or natural white pines with blister rust. There are many different kinds of resistance, so it varies. There are some trees that appear to resist the fungus. They won't even get leaf spots. But this is pretty rare. It's more likely to get seedlings that do get some kind of infection, but control or contain that infection and survive through it. There are as many as 7 different types of resistance in white pine, so varying from things like needles that get infected but become necrotic in our shed before the fungus reaches woody tissues, slow-moving infections that don't make it to the main stem before that branch dies and the fungus dies with the branch because it's an obligate parasite. There are also a variety of different what we call bark reactions, where the fungus makes it into that main stem and then the plant can control or compartmentalize that infection, and in some cases even inactivate the canker. So a lot of these improvement programs, these resistance breeding programs, they're always working toward improvement, always honing their skills. A lot of these resistance reactions are controlled by one or a few strong genes, and many of them are controlled by many genes, all contributing just a little bit. So it's very complex science. It takes time to understand and even longer to manipulate. For programs for whitebark and limber pines, these programs are still expanding. We're incorporating information and plant material from a range-wide set of collaborators. So programs like the Whitebark Pine Ecosystem Foundation and the Crown of the Continent High 5, which is the High Elevation 5-Needle Pine Working Group, all working toward improving these seedlings as well. Some researchers are exploring hybridization between white pines and Eurasian white pines. So the Eurasian pines have co-evolved with the fungus, so they do have some built-in resistance. Anytime you're doing interspecific crosses, though, it can be very difficult to maintain the horticultural characteristics of your original subject, in this case, North American white pines. So many of these programs are still exploratory or experimental. It's also of great interest to try and combine expressions of resistance or pyramid different types of resistance into individual trees. So in that way, we're creating sort of a backup or a fail-safe in case one type of resistance is overcome. Again, very difficult to understand and manipulate genetic control of resistance. So this is a long process. The biggest impact we're having on potentially restoring this species to what it once was is pairing these resistance breeding programs with large-scale landscape plantings. So pretty much all of the stock, particularly for western white pine, that's available for planting is improved seedlings, so seed from resistant parents. So by planting these seedlings out into the landscape, we're allowing this small genetic pool of improved seedlings to intermingle with wild seedlings, thereby increasing diversity and enriching that gene pool so that we can maintain adaptiveness. And also by planting these seedlings out into what we call pine sites, sites that are good for their growth, we're reclaiming these sites before they can be encroached upon by other more shade-tolerant conifers. The big hope here, the take-home, is that we're trying to get these programs to a self-maintaining, self-regenerating system so that we can truly say that we have restored white pine to the landscape. So I think that's it for me. I want to thank you for allowing me to give this talk. I hope you enjoyed it. I think it's just so fascinating, the biology of the fungus, the history of the disease. It's had such a big impact on forest health and restoration. And I really think it's one of the first events in our history that inspired a focus on maintaining our forest resources and a dedication to forest stewardship. So really neat topic.
Thank you, Katie.
Thank you.
Hey, so while I was sitting here, we just got a Science You Could News bulletin from the Rocky Mountain Research Station, and the article is predicting the future to save whitebark pine. So
right on.
Yeah, it's great. I'll put that link in our chat pod here. And if you want to pull up the Q&A window at the bottom of your screen, that can help you sort of see the 3 questions that are currently up right now. And then if anybody else has questions, please feel free to put those in the Q&A pod, and I will help facilitate them and help Katie learn what you guys want to know. But I learned a lot in the last 43 minutes, that's for sure.
Awesome.
So Glenn had a question. Can whitebark pine affect Jack Scots or lodgepole pine?
Hi, Glenn. Can white pine blister rust affect Jack Scots or lodgepole pine? No, it is specific to 5-needle pine, so strobus seedlings. Jack Scots and lodgepole are all 2-needle pines, I believe. For lodgepole, however, there is a native blister rust fungus called, I'm totally blanking all of a sudden. There is a native blister rust fungus, Commandra blister rust, which does affect 2-needle pines. It looks almost identical, and it spreads almost identically. The alternate host there is Commandra, so it's a different alternate host. But the big kicker here with Commandra blister rust is it's native. So although we will have, big years, rusts occur in wave years where there's a lot of infection, and then you might see the effects of that infection years later, and it looks like a big problem. But with these native blister rust fungi, they're going to exist in low levels. These trees have co-evolved with them, so they have some ability to resist and fight the fungus. So no, white pine blister rust does not affect the 2-needle pines, but there are other blister rusts that do.
Susan had the question about the Sulius mushroom species.
Yeah, so Sulius, I could totally be wrong. I'm going to throw it out there. I think it's sort of a mycorrhizal association, which are fungi that have a symbiotic relationship with the roots of all plants, really, but certainly trees. And I don't know the ins and outs of it, but I've certainly heard of it. And I can also suggest some people to ask about that. There are some people who are a part of the Whitebark Pine Ecosystem Foundation that work extensively with Sulius. So that is certainly something that's an area of active research, and it's pretty fascinating.
That reminds me, somebody asked if you might consider making your slides available. So I have the ability to put a link to them on our website if you'd consider that, because I think people were interested in printing out specific slides.
Awesome. I had fun making them, so I'd be happy to share them.
Good. Any special considerations for IPM for whitebark pine? You touched on a lot of those, but
yeah, at this point, hi John. Excellent. At this point, for whitebark pine, I can't think of anything in terms of pruning, I'm saying. So the pruning options, those are typically used for western white pine. For whitebark, we're exploring things like daylighting trees, so doing removal of weeds and hosts, alternate hosts around the radius of the tree to give them more resources to help them grow better. We are doing a lot of these resistance breeding programs, so identifying plus trees. With whitebark pine, because they have that relationship with the Clark's nutcracker, we actually have to cage the cones so that the birds can't get in and extract all the seeds before we get to them. So we are doing seed collection, identifying plus trees, making sure that our plus trees are ascribed to a certain climate zone and area. And then there's a big growing operation at the U.S. Forest Service Coeur d'Alene Nursery, where they grow whitebark pine seed that's been supplied to them for operational planting. So we actually have a planting doing this summer with the DNRC and the U.S. Forest Service on an old fire scar. So we're planting some of those potentially improved seedlings or seeds that came from plus trees in that area. So I think that's probably the biggest push for IPM at this point in whitebark.
Great. So Bruce asked, why do you think major gene resistance has not been discovered in white pine as it was in sugar pine?
I think it has been. I think that there is major gene resistance. I hope I'm not wrong here, but I think there's a CR2 gene that has been identified in western white pine, at least. And I think that there is some difficulty when you have major gene resistance because it is just a single strong gene. It's very easily overcome by evolution in the pathogen. So there's this, they call it there's a model of a back and forth where a tree can develop resistance and a pathogen through evolution will overcome that type of resistance. And pathogens evolve very quickly because they have so many generations in such a short time. So I think there is some major gene resistance in western white pine, but major gene resistance isn't always the best answer.
Great. I'm going to interrupt and ask Mark to launch our polls. So before you leave the meeting room, if you wouldn't mind taking our quick poll, helps us evaluate our webinar program. Feel free to drag it off the screen while Katie answers the rest of our questions in the last 10 minutes we have. So Dennis asks, I've heard of nurseries who want to sell ribes that they have disease-resistant ribes. Any thoughts on this?
I think there are some. I don't think that they are bred for resistance. Again, I could be wrong, but there's a whole range of susceptibility within native ribes. So there are very susceptible ribes that produce a ton of spores, so they're very virulent alternate hosts. But there are ribes species that aren't as sensitive to the fungus. So I'm not sure the extent to which they're resistant. If they're completely resistant, they just don't get it. So that could potentially be what those nurseries are referring to, or maybe some species of native ribes that just don't get the fungus.
Ben once was wondering if you could talk about climate change. So how will climate change affect the programs to restore white pines within their native ranges?
Hi, Ben. Gosh, climate change question. Well, I think that with anything, it's going to present a lot of problems because there's going to be a reduction of the types of sites that are perfect for white pines. There's going to be impacts on changes in plant communities. So one of the biggest encroachments to white pine are other conifers, things like Doug fir and western red cedar that are shade tolerant. So they come in, and any white pine seedlings that might get blown in don't have a chance to grow under those thick canopies. So certainly encroachment from other species. We see that a lot in our whitebark pine ecosystems too, where we have subalpine fir and Engelmann spruce encroaching and creating too much shade for regeneration of those seedlings. So I think that especially at the higher elevations, as we have changes in climate and warming and potentially drought, extended drought, this could have a huge impact on these programs. I think because at this point, some of the outplanting programs are so controlled that we might be able to choose the best sites regardless of climate change. But yeah, that's one of those classic climate change questions.
Yeah, for sure. So Carrie asked, would you mind sharing the contact information for the individuals or group involved in the Oregon BR resistance breeding program?
Sure. Oh, they are a renowned program. They are USDA Forest Service, and it is called the Dorena Genetic Resource Center. I might be butchering that, but that's D-O-R-E-N-A, Dorena. And they are the experts. Yeah, they have a lot of information available online through their website. They have pictures. A lot of the pictures I got came from their website. So yeah, I strongly encourage reaching out to them, as well as the Coeur d'Alene Nursery, who are growing some of these whitebark pine seedlings. You can give them a call and chat with them. They're really friendly folks, and they are good at what they do.
Great. So Paul Zambino had a clarification on the Western white pine CR2 gene is only found in the coastal regions, not in our inland forests. The Oregon breeding program uses both the major gene and multigenic resistance.
Fantastic. Thank you, Paul. Paul's an expert.
That's one of my favorite things about this. You never know who's listening.
Yeah.
So. Okay, great. Glenn had a question. Has there been cooperation between white pine blister resistant tree research and mountain pine beetle resistant tree research that you know of?
That's a good question. I am familiar with one paper where there was apparently bristlecone pines that were observed that are resistant somewhat to mountain pine beetle. So that was a pretty interesting development. I don't know that there's a ton of, and I could be wrong, but I'm not familiar with the extent or the depth of mountain pine beetle resistance research. So I think that since it's a native beetle, we don't typically regard it as something that we typically need to protect against. I think there are a lot of factors that led into the latest outbreaks that occurred throughout the 2000s here in the West. And that's a whole conversation with a lot of opinions. But I think that there is a resistance that has been observed in certain species to mountain pine beetle, but I'm not sure the extent to which the actual research.
Has there been any research on chemical control for the fungus that you know of?
I don't know, actually. I don't. I'm trying to think. In terms of things like bowl injections or things like that, I'm not familiar with anything. And because it's in such a way that it's distributed over 2 hosts, you really have to determine which host you're going to target. So for a high value white pine, something you want to save, I'm not sure. There may be some kind of chemical controls. But in terms of limiting, if you still have both hosts, you would need to almost target both hosts to have any kind of reduction in disease occurring. So I'm not sure.
And then Paul Zambino chimed in again. There are major genes for resistance in many native ribes species. The European black currant ribes,
Right.
Nigerium. There are some cultivars that have a gene for resistance that has been overcome by the fungus in the New England states.
Thanks, Paul.
Awesome. Let's see. Jessica Shaw asked if the recording and slide presentation will be made available. Yes, they're all available on our website, our Utah State University Forestry website. So if you're subscribed to our newsletter, there's always links to that website in there, and that is a quick, easy way to find them.
Cool.
I think we've got all of our questions answered and addressed. So thank you so much, everybody, for chiming in today. Please take our quick poll. It helps us do our job better. If you need CEUs, follow the link to that Google form and fill it out completely, and I'll make sure that CEUs are submitted. Thank you to Katie so much for making this great slideshow. Really appreciate it, and it's really informative. And send me your slideshow, and I'll put it on our website if you wouldn't mind. And thank you, Mark.
You're welcome.
Thank you, Mark.
Signing off. Everybody have a good day.
Okay, bye-bye.
Bye.